Salam everybody. I'm going to explain this, the third component of connective tissue, I mean ground substance. The ground substance is a hydrated and transparent part of extracellular matrix. And also it has three major macromolecules. And these are GAGs, acronym for a glycose aminoglycans, protoglycan or PG, and GP also for a glycoprotein. These are components of ground substance. The example for them, GAGs like hyaluronic acid, or called hyaluronan, chondroitin 4 and 6 sulfates, dermatan sulfate, keratan sulfate, and heparan sulfates. These are GAGs. About the protoglycans, you see the protoglycan consists of protein plus GAG. It means the GAGs aggregated around or attached around the protein and make a protoglycans. Among the GAGs, except the hyaluronic acid, other GAGs aggregate around the protein and make a protoglycan. Because of that, you see these GAGs also attached to the protoglycan. It's a subgroup of protoglycans. But hyaluronic is a pure GAGs. And these GAGs attach to the to the proteins and make a protoglycan like aggrecan, syndecan, and decorin. And example for glycoproteins like fibronectin, laminin, and chondronectin. If you remember, we had laminin in a basal lamina or basement membrane. It's kind of glycoprotein. Let's start with GAGs. The definition for GAG is, the GAG is a polysaccharide. Poly means multiple, it's many, a lot. Saccharide means sugar, carbohydrates. It means the GAGs consist of many sugars, consist of many carbohydrates. I show each carbohydrate or each sugars here as a hexagon. So these polysaccharide, I mean the GAGs, is consist of repetition of one disaccharide. Disaccharide means two sugars. I again showed disaccharides here with two hexagons, one in blue and another in red, and you can see the repetition of this disaccharide. So, in the GAG, a repetition of one disaccharide make a polysaccharide. We call these polysaccharide GAGs. Another characteristic for the GAGs is a linear polysaccharide, not branch polysaccharide. You can see here a branch, have two branches of sugars. But the GAGs is a linear polysaccharide, just repetition of disaccharide in a linear pattern. For example, when we have these two carbohydrates, for example, glucuronic acid or N-acetyl D-galactosamine, these repetition of these disaccharide make a chondroitin sulfate. When we change the first sugar, for example, replace it with hyaluronic acid, the second one is same, the name of the GAGs change. It gets dermatan sulfate. Or we change the second one, make a heparan sulfate. If you remember again, we have heparan sulfate. in a basal lamina. I don't want you to memorize the name of this sugar. Just we have a sugar disaccharide, and we don't want the name of the exact name of each disaccharide. Again, with another picture, you see hyaluronic acid or hyaluronan here. We have one disaccharide, and repetition of this disaccharide make a polysaccharide and a linear polysaccharide. We change the second sugar, we have another GAGs. Again, we change the first, we have another dermatan sulfate and others. You can see here the hyaluronic acid have a difference with others. You see other GAGs have a sulfate group, but the hyaluronic doesn't. Because of these sulfate groups, these GAGs can attach to the protein and make a protoglycan, and the hyaluronic again doesn't. It's a pure GAG, but other make a protoglycans. And the hyaluronan or hyaluronic acids among the other GAGs, and unlike the other GAGs, produced by an enzyme on a cell membrane. It means the hyaluronic acid is synthesized by the enzyme on a cell membrane directly out of the cells, into the extracellular matrix. It doesn't produce. It isn't produced by a cell inside of the cells. But other GAGs are synthesized inside of the RER and completed in a Golgi, and then cells secreted it outside of the cells by exocytosis. But the hyaluronic acid directly synthesized out of the cells. And another characteristics for GAGs is they have a lot of a negative charge on the sugars, on the carbohydrates. For example, sulfate groups, carboxyl groups. We said they have a high content of negative charge. In our body, for neutralize this negative charge absorb a positive charge. The most abundant positive charge in our body is a sodium. So the sodium attached to this negative charge for neutralize it. And accumulation of the sodium around a GAGs make an osmotic concentration. So absorb water and make a structure like a jelly. Due to this characteristic, we call the GAG linear mucopolysaccharide. Muco means like a jelly, mucus. And also, this negative charge have another make another properties for GAGs, and that is, the negative charge or the same charge usually repel each other. So cause the GAGs have a linear or have a extended conformation. they never coil. Today's in medicine we use hyaluronic acid for cosmetic use. We can smooth out the wrinkles in our skins. You know, today the men also use this medicine or this material for get beautiful. We have a table here. We have the name of gags and the site of their distributions. You should know the site of distribution of these gags in our body. For example, hyaluronic acid we have in umbilical cord, synovial fluid, and vitreous humor, and cartilage. As you know, the vitreous humor is like a jelly because it has a high content of hyaluronic acid. Let's go to the explain protoglycan and glycoprotein. Both of them consist of protein plus carbohydrate or glycans and make a protoglycans and glycoprotein. Both of them are compound noun from protein and glycan. In the first one you see the protein is incomplete, we don't have iron, and glycan is complete. In the second one is vice versa. It means in the protoglycan, the glycan is complete, so we have high content of carbohydrate. And the second one, the protein is complete, it means we have a high content of protein and less glycan here and less protein here. Let's see by pictures. In these two pictures, in the first one here, I mean in the left, this color means the protein and the black is carbohydrate. In this one also, this is protein, purple one is protein, and green are carbohydrate. Which one is glycoprotein and which one is protoglycans? This one, yes, it's a protoglycans because we have many carbohydrates and just one protein. And here we have a huge and big protein and a few carbohydrates. It's a glycoprotein, it's a protoglycan. And if you remember, I said the protoglycan consists of protein plus GAG. You can see a lot of GAG. Why I say these are GAGs? Because they are polysaccharide, linear polysaccharide. You can see all of them are linear, not branched. So this protoglycan is glycoprotein. You see the protoglycan is like these tools, tube cleaning brush. It's very similar to this. If you remember these tools somewhere, it should memorize you the structure of protoglycans. But in the extracellular matrix, under the electron microscope, we can see, if we fix very well, we can see the GAGs and protoglycans. Usually, the large carbohydrate, large GAGs, I mean the hyaluronic acids, take place at the center or located at the center and accumulate other protoglycan around of itself and attach them by a linker protein. Magnify this part, you can see the hyaluronic acid attached to the protoglycan, protein, and GAGs by this linker protein. The exact protoglycan are aggrecan, syndecan, and decarin. Let's see them by their shape. You can see here the extracellular matrix, fibers, hyaluronic acid, and a lot of structure like that brush I showed you. These are agricans. You can see a lot of agricans attached to the hyaluronic acid in the extracellular matrix, and they fill the space between the cells and fibers. So they also absorb water and fill this space with also water. About the syndecan. Syndecan is not a proteoglycan in extracellular matrix. Instead, it's the proteoglycan on a cell surface. You see the cell membrane, an integral protein on a cell membrane attached to some gags. And you know the gags can absorb water. So by the water, they can absorb a lot of growth factor. For example, here, FGF, fibroblast growth factor. And also you can see the structure of decorin. It's a protein with a few gags. Here the decorin role is to attaching the collagen fibers to fibrils together and make a collagen fibers. Helps the collagen to make a fiber. About the glycoprotein. You can see two glycoprotein here, laminin and fibronectins. We have a laminin in a basal lamina, and fibronectin also we have a basement membrane and also in the connective tissue. Laminin attach the cells to the extracellular matrix, or is the site for cell attachment. Both of them are the site they are a site for cell attachment. For more explanation, let me see a picture. I usually said the proteoglycan, the glycoprotein is like a triangle, something like this. And each angle is site for attachment of each connective tissue component. For example, this angle is for attachment of cells, this part for fibers, and this part is for a other gland substance in glycoproteins. Because of that, we call the glycoprotein multi-adhesive glycoprotein. It means they have a multi-place for attachment. For showing this attachment site, usually show the glycoprotein with different color. You can see blue, dark blue, light blue, and also green. For example, this part for a cell attachment, this part for a gland substance attachment, and this part for a fiber attachment. Also in the fibronectin, also you see different colors. It means they have a multiple site for attachment. You see, we have some tools like this. That's like this glycoprotein, just for memorize them. Laminin is, let me clean my draw and back to the arrow. You see the laminin is like a cross, and the fibronectin is like a nunchago. Usually the cells attach to the glycoprotein by an integral protein on their surface. Usually the integrins. Integrins are protein on a cell membrane. The cell can attach by them to the glycoproteins. Integrins have two parts, or have two chains. alpha chain and beta chain. Because of that we call them heterodimer protein. Have two different parts, and you see this protein from the inside of the cell attached to the cytoskeleton, and from the outside it's attached to the extracellular matrix, especially on glycoprotein. This picture also shows the cell membrane, integrin, have two parts, attached to the glycoprotein, and the glycoprotein from the other side attached to the to collagen and to the proteoglycans. So the glycoproteins can attach cell to the extracellular matrix. Without this glycoprotein, the cell cannot attach to the extracellular matrix. And the last part for the ground substance is the water inside of the ground substance called interstitial fluid or tissue fluid. This fluid comes from the blood plasma. You can see an arteriole here. Arteriole means a small artery, then change to the capillary and change to the venule. Venule also means a small vein. Two forces also worked on the underwater of the capillary. The first one is hydrostatic pressure. The second one is osmotic pressure. The hydrostatic pressure caused by a heart pumping. Heart pumping force the water inside of the capillary goes out, force the water out of the capillary. For example, for learning, 10 cc comes out. And when this blood comes to here, because comes to the venous, because the big protein cannot go out by this pressure and remain into the blood and make osmotic pressures, so drown back the water inside of the venous. For example, 8 cc down back, 10 cc force out, and remain 2 cc inside of the connective tissue. And these 2 cc drain continuously by another vessel called lymphatic vessels, lymphatic capillary. So by this way, our blood nourish the cells of connective tissue. By 10 cc, the nutrients comes out. By 8 cc, the waste material drawn back into the venules. So the cell nourishments happen by this way. Every disorders happening here, for example, the cancer cells blockage the lymphatic capillary, cause accumulation of excess water in the connective tissue and create the edema. Okay, we finish the ground substance in connective tissue. In the next movie, I will explain the types of connective tissue.